All right, excellent. So today is Tuesday, September 21st. We are going to start our discussion of our immune response, talking about our body defenses and how we protect ourselves from all the big baddies and little baddies that are out in the outside world. Today will be the last day. I'm hoping to finish the lecture at least an hour early, if not a little bit more, so that you guys can have some class time to work in your groups. Many of you took advantage of that. And again, once we break into groups, if you want to all leave and go meet at Starbucks instead, you're welcome to do that. You're not required to stay during that time. However, I do believe it is important to have this time today. So if there are groups that are struggling finding information, or if you've realized that you absolutely hate your topic and want to change it, today would be the last day to be able to change your topic. So speaking of topics, every group except one, group 6, and group 6, you know who you are. Everybody else has posted their names and their topic in their discussion board. So group 6, please remember to do that. But like I said, if you're having struggles finding it, or you don't like your topic or something along those lines, this would be the last day to make that change. And then if there's any questions or concerns that you guys are having, this would be a perfect opportunity to bring those questions up with me as well. To avoid the confusion that we had on last class, again, Thursday this week, we do indeed have class. We are meeting. We're going to continue our discussion of our immune response. Then there's just no assignments due, because there's less assignments in this section, because I figure you're working on your group presentations. That doesn't mean there aren't any assignments. There are 2 more assignments. You have one PhysioX activity, again, just activity 3 of exercise 12 that you'll be doing. And you have a Labster, at which, again, remember, need to be 100% complete and 80% correct. Again, one of the few advantages of being online is we can do things a little bit differently towards that end. Remember, we are turning in our outlines by Friday. Again, if you complete it ahead of time, don't wait. Turn it in so that you don't forget. But I do want to give as much time as possible for people to continue to work on these projects. And at the same time, I want to make sure that everybody has the weekend to look at everybody else's outlines in preparation for the presentations that will be on Tuesday, because this is testable material and that lecture exam is going to be on Thursday. So sitting through 9 presentations on Tuesday is a lot of material to have to try to retain for an exam 2 days later. So having those outlines ahead of time can help people to start looking at some of the key points and the key highlights and the key concepts that are going to be covered on those presentations. So that group outlines need to be turned in by 6 p.m. on Friday the 1st. Make sure you have everybody's name in your group on that. Remember to have your topic, and remember to also have your references. Remember, if it doesn't have the references, or if it's not turned in on time, you will be penalized severely on those. So make sure you do that properly. All righty, questions on any of that? Oh, wait, hold on. Yes, so I just need 1 outline per group. That is correct. So I don't need 3 copies of the exact same outline from the 3 different people that are in the group. I just need 1. Each group is responsible for 1 outline. So 1 person in your group should be responsible for turning that in. And then there should be 9 outlines that will be posted onto Canvas, because we have 9 groups. So yes, 1 outline with everybody's name on it is all that's necessary. All right, because the outline, again, it's not just the outline of your part. I want an outline of your presentation. So it's a group thing. All right. Well, and actually, I think, hold on, now that I think about that, I believe yeah, it is set up as a group assignment. So you'll be putting it into a folder for your group. So yeah, set up as a group assignment. So yes, 1 person, and then you'll get that for everyone. All righty. Any other questions? All right, let's dive into the next major topic we're going to be talking about. We are going to be working our way through the immune response and our body defenses. There are a couple things that we'll be working on for that. Obviously, if we're going to protect ourselves, we have to have an understanding of what we're protecting ourselves from. So I will take an entire microbiology class and distill it down into 10 minutes of information. Everything else, after this 10-minute discussion, you will no longer have to take microbiology, because I'll teach you everything you need to know in that 10 minutes. And everything else they're saying is pretty much stuff they're making up anyway. We will then talk about our body defenses, both the specific and the non-specific, or our hammer and scalpel defenses. And then you will be presenting us with the information about our immune disorders and our immune deficiencies. So that's what you'll be doing in our group presentations. All right, as I mentioned, let's talk first about what we're trying to protect ourselves from. Again, the world is covered with microorganisms, massive, massive amounts of them, right? A square centimeter of your skin has something on the order of 10,000 bacteria on it. Now, of course, when I say that, usually people go diving for their Purell, but do you necessarily want to dive for your Purell to get rid of all those 10,000 bacteria that are on the surface of your skin?
No.
No, because they're actually resident bacteria. They're actually benign. They're in some ways beneficial. So many of the microorganisms that are on the planet, we happily coexist with. However, a pathogen is a microorganism that can cause damage to us, can cause diseases, other disorders, and in extreme cases can cause death. And of course, what are the most common types of pathogens?
Bacteria and viruses.
Exactly, they're bacteria and viruses, absolutely. These are by far the most common. Oops, oh, it's going the wrong direction. There we go. Sorry. All right, let's talk about bacteria. Bacteria are single-celled organisms. They're prokaryotes. Remind me again what that means?
No nucleus?
No nucleus. And not only no nucleus, but actually no membrane-bound organelles at all. Right? So there's really no specialization. It's just a bunch of hunk and junk thrown into a plasma membrane, and poof, you have that bacteria. They are incredibly simple in structure. They come in simple in shape. However, they are also the most effective organism on the surface of the planet. Now, typically when we talk about bacteria, we talk about the bacteria that are responsible for causing diseases, like E. coli, like botulinum. Someone remind me again what botulinum is?
The toxin.
It does produce the botulinum toxin. Where does botulinum bacteria typically grow?
Canned food.
Yeah, inappropriately canned or jarred foods, absolutely, that have spoiled, right? And what kind of condition does it cause? That toxin formed by the botulinum bacteria.
Paralysis.
Yeah, it causes paralysis, absolutely, right? It's the problem with that is that it causes a breaking of a protein that plays a role in our neurons being able to release neurotransmitter, so we can't talk to our muscles anymore. Right, so you can, again, lose the ability to breathe, lose the ability to move an arm, right? Botulinum toxin sounds super, super scary, but if we give it a little cutesy name like Botox, then people have no problem with them injecting it all over your body, right? To loosen the muscles of your forehead so you no longer have wrinkles, or inject it into the sweat glands in your armpit so you no longer have to produce sweat, right? So because there's nothing worse than having a stinky armpit. So it's better to risk the chance of getting that botulinum toxin into your blood and stopping your breathing than showing the fact that you biologically produce sweat, because as we know, it's only on the outside what matters, right? Although let's talk about this one here, Yersinia pestis. Anyone have any idea what bacteria this is commonly referred to as? Anyone taken micro yet? Come on, there's got to be someone in there that's taken micro or is taking micro. What's Yersinia pestis commonly known as? I'll wait if you have to Google it.
Is it the plague?
Not just any plague, but the Black Plague. Absolutely. Yersinia pestis is the Black Plague. Since the invention of agriculture, the population of humans on this planet has increased. It's gotten to the point where it's almost increasing logarithmically. Right? So once we learned to plant food and be able to produce more food than we needed, the number of births have outweighed the number of deaths in the entire history of man, except twice. Only 2 times in the history of man have the number of deaths outnumbered the number of births in the world. And both of those instances, it was caused by this bacteria, Yersinia pestis, the Black Plague. All right, so again, while these can be incredibly simple, they're also the most ubiquitous of all of our microorganisms. All right, they're the most effective organism on the planet, somewhere around 5 towards 10 to the 30, and I have no idea what real number that is, number of bacteria on the surface of the planet. So again, incredibly timely, incredibly simple, but have the potential to be incredibly powerful as well. Viruses, as we've all got to know very, very well over the past couple of years, are a different type of infectious agent. They're minuscule, 1/100th of the size of bacteria. And when you take microbiology, you can get into this great debate as to whether or not viruses are even alive. If you think back to our general biology, when we talked about what it means to be alive, how can we tell the broccoli is alive and the rock isn't when they both just sit there, right? Because the broccoli is able to do things like metabolism. It's able to do things like grow, respond to its environment, all of those types of things. Well, a virus isn't capable of doing those things. It's incredibly simple. Most viruses are just a simple protein coat on the outer surface containing some type of genetic material inside. And when it's just sitting here on the counter, it's not able to do any of those things we associate with life. However, once it's introduced to a host, it can do those things. So like I said, you'll have a fun day in micro debating whether or not viruses are alive. I don't care. But we've taught when we care about viruses is when they cause harm, AIDS, rabies, hepatitis, viruses called cold, flu, warts, and your friend and mine, COVID-19. And all these are examples of viruses that have the potential to do harm. Now, when evaluating a pathogen, and this is one of the things, again, all these are things we've learned a lot about over the past couple years. Oh, I realized my microphone's not plugged in. You'll have to tell me if I sound better now. When evaluating a pathogen, there are 3 basic characteristics we look at to see how dangerous, how much of a health risk it produces. The first of those characteristics is the mode of transmission. How is it transferred from one person to another? All right. This was one of the big issues, big questions that was being asked about COVID-19 when it first came out. Is this something that can be airborne, or is it something that is in the larger droplets, past mucous membrane to mucous membrane, right? So is it something that can get into an air conditioning and be spread throughout an old folks' home? Or is it something that basically you have to share breathing in large water droplets with, right? Or is it something that comes from the exchange of bodily fluids, all right? Things along those lines. Related to this, but not identical to it, is transmittability. Transmittability is how easy is it to pass from person to person. Obviously, something that requires the exchange of blood between individuals versus something that is airborne. The thing that is airborne is much more easily transmittable. However, it's more than just the mode of transmission. Something that is airborne, right, may only be viable outside of a host for 2 seconds, in which case you would pretty much have to breathe in someone's face to get it. Or something could live on a surface for 2 days, in which case if I cough on my hand and then as I'm leaving the classroom, I touch the doorknob, and then an hour later you leave and touch the doorknob, right? Is that virus still transmittable at that point? So obviously the mode of transmission affects and influences how easy it is to pass, but it's not the only factor. Like I said, it could affect how long is it viable outside of the body, how easy is it to spread from person to person, things along those lines as well. And then, of course, the third factor that determines how harmful a pathogen is its virulence, how much damage it can cause by that infection. The common cold is airborne. The common cold virus can last on a surface for hours. So it has a very easy mode of transmission, a very high transmissibility. But how virulent is the common cold?
Very low.
Very low, right? It makes most people feel like crap for a couple of days, and then they recover. Right? People who are immune compromised or may have other issues, very old, very young. It's not like it's completely incapable of hurting someone, but most common healthy adults in particular, it's not very virulent. Ebola, on the other hand, has an 80% mortality rate. Right? So when you get Ebola, 80% of the time, you're going to die in less than 2 weeks. So why isn't Ebola spreading around the country the same way COVID-19 is?
I'm not sure, but I'm guessing it's not transmitted like as small of droplets, or like it not as transmitted as easily.
Exactly. In fact, Ebola requires blood-to-blood transmission. Again, that's why Ebola outbreaks typically occur in hospitals, where you have a lot of sick people and you have a lot of doctors and nurses who are helping those sick people and then helping someone else who's not sick. And so typically when an Ebola breakout occurs, it occurs in that type of a setting, a war setting or a hospital setting, because it requires that blood-to-blood transmittability. Yes, also, one of the things that affects the transmittability is because it kills off the individual so quickly, they don't have as much of an opportunity to spread it because time-wise that way as well. And it's hard as a less susceptible mode of transmission, right? Right. Again, before COVID, one of the things that would come out every couple of years was one of these great pathogen-related, horror movies like Outbreak, right, which starred that monkey from Friends, where something like the Ebola virus becomes airborne. Right, you have something that's very, very virulent that gains a more easily mode of transmission, which makes it more transmittable, which makes it much more dangerous. All righty, questions on that? All right, so there, like I said, now you don't have to take micro anymore. I've explained it all to you, everything that ever needed to know about micro and more. But now that we've got a basic idea that there's a lot of scary stuff out of there, the good news is we have some pretty good body defenses to be able to help us with that. Now, as I mentioned, there are two types of defenses. One of those defenses is a non-specific or innate defense. This is a rapid response that helps to protect us from many different types of invaders. This is what I like to lovingly refer to as the hammer defense, right? As a wise man once said, if the only tool you have is a hammer, then the whole world is a nail, right? Obviously, if you need to hang a picture frame, you can grab that hammer and hammer in a nail, or you can hammer in that screw. Leaky pipe, hit it with your hammer. Right? That hammer is used for everything. It's quick, it's fast, there's no thought gone into it. You've got that one tool, you know how to use it. So if you see a problem, you hit it with the hammer. But what it lacks is specificity. Specificity is our specific or adaptive defense. This is often what we refer to as our immune response, right? Our non-specific, our hammer defense, it doesn't care whether something is a bacteria or whether something is a virus. We're going to hit it with a hammer. With our specific defense, not only do we care if it's a virus or a bacteria, but we care whether it's virus A or whether it's virus B. That gives us tremendous specificity. That gives us tremendous precision. And so this is our scalpel defense, right? Where we have that precision. Rather than just hitting with a hammer, we can cut out what that precise problem is. Now, innate and adaptive, specific and non-specific, are two perfectly acceptable ways of talking about these two defenses. But the other way to talk about this, and actually, hopefully after the cardiovascular system, you've spent some time with that interactive physiology. If you haven't, then I would strongly encourage, well, I know you've at least done one assignment because I made you do it for a grade. Your interactive physiology has this amazing tutorial where it talks about our body's defenses, and it uses the analogy of protecting a castle. We have this castle that needs to be protected, and we have these two types of defenses, innate or adaptive, right? Non-specific or specific. But we could also think of these as our three lines of defense. Our first line of defense, hold on, let me move this out of the way. The goal of the first line of defense is keep bad things out. This can involve things like surface barriers, secretions, other specializations that help to keep bad things out. Because if the bad thing can't get in, then it can't cause any harm. Our second line of defense, our goal is twofold. If something is able to penetrate our defenses, we want to limit the spread and we want to limit the damage. We're going to do things like use phagocytes, use inflammation, use fever. And notice in both of these they're the same. It doesn't matter whether it's a virus, whether it's a bacteria, whether it is, right, a burglar, whether it is someone with some type of religious pamphlet that wants to get into your house and talk to you. Whatever the reason, whatever the goal, it doesn't matter what it is. We either want to keep them out, or if they get in, we want to limit the spread and limit the damage. So notice these are both parts of our non-specific defense. But ultimately, if something gets in, then we're going to use our specific defense. And our specific defense really has two major goals. Goal one is to precisely defeat that pathogen, destroy whatever that harmful thing is, and provide protection for the future. To protect us if we ever come in contact with that bad thing a second time. So we can also take these two lines of these two types of defenses and divide them into these three lines of defense. And I think in some ways, it's an easier way to think of these things. Let's do this. Let's talk first about our first line of defense, part of the non-specific defenses, with, again, that goal of keeping things out. So let's switch to our whiteboard. So again, we're talking about our non-specific defenses. We are talking about, in this case, our first line of defense. And again, remember, the goal of our first line of defense is to keep bad things out. So give me some examples of some of the structures that we have in place that help to provide that first line of defense to keep things out?
Hair.
Skin.
Okay, hair, excellent. Let's be more specific. What did hair wear for starters?
Like your nose.
Nasal passages.
Excellent. So nasal. Oops. Anywhere else?
Ear canal.
Let's be specific. Auditory, external auditory meatus. Which is a fancy way of saying our ear hole. Excellent. And what do those hairs do?
They grab incoming pathogens or any other things that they basically prevent entry.
Yeah, exactly. They prevent entry of large dust and debris. That large dust and debris is then able to stop smaller pathogens from being able to get in, stop insects from being able to get in, large bad things that way. Absolutely. Give me another example.
Skin.
Skin. Excellent. Skin does lots of things. Tell me some of the ways that skin provides this first line of defense.
It's a physical barrier.
Excellent. It forms a physical barrier. A very successful physical barrier. How? What is it about skin that forms that physical barrier?
Multiple layers.
Multiple layers, right? Multiple layers of what?
Tissue.
Well, not so much tissue. Oops. Multiple layers of what?
Cells.
Cells. There you go. That form a tissue, and what tissue in particular forms the superficial layer of our skin?
The epidermis.
There you go. Amy's got it. Keratinized, stratified, squamous epithelial tissue. So we have many cell layers. And what do we know about some of those cells?
They're dead.
Yeah, absolutely. The superficial layers are dead. Why are they dead?
Because they continue to regenerate underneath and push.
Well, that's
not
why
push,
we're not
but-
naked. Absolutely. Because even though they're dead, we're able to make more to replace them. But why did they die? Is it the fact that they're dead what causes the protection? There we go.
Is it
Amy's
blood
got it.
supply?
They are
Oh.
filled with keratin. Keratin, remember, is that hard protein that forms that tough outer layer. And remember, keratinocytes also produce those lamellated granules that basically fuse and waterproof the layers. So we have this big, thick, hardened, waterproofed layer on the outer surface of our skin. But there's even more than that. Someone pointed out, I'm sorry, I didn't catch who said it. We are constantly making more. Why do we have to keep constantly making more?
Because they flake off.
Exactly. And when they flake off, what do they take with them?
Whatever was hanging onto them.
Right. Remember, we talked about those resident bacteria. We have that resident bacteria that are able to basically outcompete some other harmful bacteria. So some harmful bacteria comes and lands on my arm. There are thousands of bacteria already there that are competing for the nutrients, the resources, and that poor little harmful bacteria isn't able to get any. Of course, if I'm worried about COVID and I completely bathe myself with antibacterial stuff, then it's an even breeding ground where that harmful bacteria comes in. It may be able to find purchase. It may be able to start to grow. But if the cell that it's living on sloughs off, it takes that harmful bacteria with it. So that combination of not just the hard outer barrier, but the fact that we're constantly shedding it and we have that resident bacteria on top are great ways that our skin provides protection. And it even goes more than that. What else does our skin do? I know it's been a long time, 430, since we talked about the integumentary system, but I can think of at least one more good thing that our skin does to protect us.
Produces sebum, which is
Excellent. What does sebum do for us?
it's acidic. It provides an environment that pathogens can't grow on.
Yep, it's a hostile environment for some bacteria. It actually has an antibacterial quality to it. And what else does that sebum do for us?
Does
What
it
happens-
moisturize the hair?
Not just the hair, but the skin as well. Think about it. If you ever had a job like working with food where you're constantly washing your hands, as you're washing that sebum off, what starts to happen to your skin?
Dries.
Dry,
It dries, and when
cracks.
it dries, it can crack. Absolutely. So it helps to maintain the integrity of the skin and the hair. Excellent. So our skin plays this huge, huge role. What else?
Temperature,
Say again?
temperature.
True. I mean, that is something that our skin does for us, but I'm not sure that necessarily helps in playing a role in maintaining the outer surface of our body. When things come inside, temperature is going to be a huge deal, but not so much for keeping things out. Temperature is not changing our temperature isn't part of the first line of defense. I think we've done everything we can done with the skin, but are there other ways that our body protects us from harmful outside things? Let's go back to that ear hole. Anything else in that ear hole besides hair that helps to provide protection?
Earwax.
Earwax.
Earwax. Of course, being sophisticated anatomy and physiology students, I'm not going to use such a crass term as earwax. I'm going to use the appropriate anatomical term for it. And of course, what is that?
Cerumen.
There we go. Cerumen, produced by our ceruminous glands. Again, it is antibacterial. It plays a role in helping to retard insects. It's an insect repellent, things along those lines. What else keeps things out? Mucus, excellent. Like where?
The nose.
Excellent, absolutely. That mucus in the nasal cavity captures dust and debris. Excellent. What else?
In the lungs.
True. Well, though, really, do we want the pathogens to get all the way down to the lungs? No. So we have mucus not just in the nasal cavity, but also in, for instance, our trachea, which is going to play an important role in helping to get that mucus elevator with that ciliated pseudostratified columnar, capturing that dust and debris and moving it up. What else? Think of some of the other openings that we have to the outside world and how we protect them.
Vaginal canal.
Absolutely. So one of my absolute favorite things to say in this class is that the vaginal canal is an incredibly hostile environment, right? And again, that is fun to say, but let's be serious about why that is so vitally important. One of the things, and again, we'll talk about this much more when we get to the reproductive system, but one of the huge differences between males and females is the male reproductive system is a closed system, meaning basically it is completely contained tubular structure. So for instance, if something harmful gets into the male reproductive tract, it stays in the male reproductive tract. The female's reproductive tract is an open system. It is actually open to the abdominal pelvic cavity. So a harmful pathogen that gets into the female reproductive tract can not only just affect the reproductive organs, but it can actually get into the abdominal pelvic cavity and affect many more organ systems as well. It's one of the reasons why sexually transmitted diseases are so much more dangerous for females than they are for males, because of the openness of their reproductive tract. And since the vaginal canal is the opening of that reproductive tract, it is vitally important that the female protects herself that way. And there are plenty of protections in there. There is the hymen, which helps to partially close the vaginal canal. So before they become sexually reproductive, to limit what can get inside. There's the mucus plug of the uterus that helps to, again, limit the materials that can get inside. And the glands of the cervix produce a very glycogen-rich secretion, which resident bacteria in the vaginal canal are able to digest, producing lactic acid, making it a very acidic condition, acidic environment. It's actually called the acid mantle. That helps to play an important role in providing that protection to, again, help protect the female from the dangers of the outside world. Excellent, 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 excellent. What else?
Tears.
There you go. Tears, absolutely. Obviously, we need to maintain not just the moisture of the corneum and the sclera, but the tears also have antibodies in them. They have antibacterial components like lysozymes. Now, lysosomes, remember, are organelles inside of a cell, but these are things that help to attack and destroy bacteria. And I think there's one more obvious one you guys haven't talked about yet. Two, actually.
Saliva.
Yeah, there we go. Saliva also has antibodies and lysosomes. Pardon me, lysozymes. In them that are going to provide some protection. Although it doesn't necessarily have a lot of acidity in there, but luckily our mouth with that saliva is located adjacent to what? True, the tonsils, part of our immune defense that way. I was thinking a little lower. Where do things go from the mouth?
Esophagus. Stomach.
And then into the stomach. And what's so special about the stomach, Amy? Or anyone?
Healthy bacteria. Very acidic.
There you go. Stomach acid, absolutely. What's the pH of your oral cavity?
Is it around 7?
Yeah, it's around 7, pretty neutral. What's the pH of your stomach?
Was it 1?
Yeah, 1 to 2. So it is incredibly acidic. So that stomach acid plays a huge role in being able to destroy any harmful material. Think back to that mucus we were talking about. Your nasal cavity, your trachea, they collect mucus and both of them move it to the back of the throat, where two things can happen. You can either spit it out, get rid of it that way, or you swallow it into the stomach, where that stomach acid is most likely going to destroy it. Excellent. Those are the obvious ones I thought of. Can anybody think of anything else?
So
I'm not
would,
sure, but oh, what about like the rectum? That's the only like where we haven't discussed like.
Excellent. So let's talk about the rectum. Are there necessarily rectal secretions? Not really in the same
No.
way. No, but it is where substances come out as opposed to coming in. What kind of tissue type is located in the anus? Same as the vaginal canal, same as the oral canal.
non-keratinized, simple squamous or stratified?
Simple? Yeah, there you go. It may not have the keratin, but it still has many, many, many layers, again, providing that protection, not just from the friction and from the expulsion and the things that are going on there, but also to provide many layers of protection that way as well. And then, of course, the rectum feeds into the colon. Is the colon one of those places that is completely sterile?
No.
No.
No, as we talked about, once again, we have our own resident bacteria in there, including things like E. coli. E. coli can be a very potentially dangerous bacteria, but as long as it's contained within the large intestine, housed in there, it's actually beneficial, it's helpful. So yes, some bacteria may be able to get in that way, but remember the colon is specialized to maintain that bacteria in there. I think we have someone doing celiac disease where we can talk about some conditions where it's not able to fully contain those things and the problems that can come along with stuff like that. But normally it's specialized for containing those. So again, it limits the ability. Remember, as we talked about, the lumen of our digestive system is outside of our body. So yeah, by keeping those things in there, that provides that protection as well. Yep. Excellent. Anything else?
Yeah, lysozymes, are those like cells that,
No, those are enzymes, proteins that help to break down bacteria that are in secretions like our tears, like our saliva, that help to, again, break down the bacteria before they ever get into our body.
So would dendritic cells be an adaptive defense?
Great question. They are not adaptive, but if it's gotten into our epidermis, then it is part of our second line of defense. It will have already gotten into our body, and that the point of that is to then limit the spread and limit the damage that it could do. So yes, there are going to be those dendritic cells of our skin are going to play an important role in providing the defense, but they're part of our second line of defense. They're something we need once it's already gotten into our body. Notice the point of all of these are to keep them outside of our tissues, outside of our body. All right. Like I said, I think those were the obvious ones I was thinking of. There might be one or two more that we haven't thought of, but I think that's definitely a pretty good list. I'm sure your book talks about more of them. Basically, if you think about it, they all fall down into things that are mechanical barriers or chemical barriers, things that are either physically or chemically keeping things out or destroying them. We talked about lysozymes. Oh, I spelled it wrong with the S, which, as I mentioned, are an antibacterial enzyme. Your tears and your saliva also contain defensins, which are also an antimicrobial peptide to help to break things down as well. So again, those are examples of some of the chemical barriers that we will see to help with that. All right, so that is our first line of defense. Again, part of that non-specific defense. And again, the goal is to keep things out. Questions on that? All right, then let's talk about our second line of defense. Let's cheat and go here again just to maintain consistency. Again, this is still part of our second line of our non-specific defenses. This is our second line of defense. And as we mentioned, the goal here is if it's able to penetrate our barrier, if it's able to get into our body, we want to limit the spread and limit the damage. Again, this is still a non-specific defense. If you think about it, that mucus in your nose doesn't care whether it's a virus or whether it's a bacteria. Right? It's the same thing true here. The second line of defense doesn't care if it's a virus or it's a bacteria or if it's a parasitic worm or anything else. These are going to be, again, general, non-specific types of defenses. These include our phagocytes, our eater cells. And someone remind me again, what are the two main types of leukocytes that become phagocytes?
Neutrophils and monocytes.
Lymphocytes.
Excellent. Neutrophils and our monocytes. Again, most of our white blood cells have some phagolytic properties, but the neutrophils and our monocytes are the two primary ones. And in fact, the monocytes, when activated, become our big eaters, those macrophages. We also have cells that play a role in our immunological surveillance. These are those night watchmen that are roaming around with their flashlights, checking out the museum, looking for bad and harmful, scary things that are going around in there. But it isn't just cellular. Again, as I said, one of the big things about this process is that it's cellular and it is also chemical as well. There are many different types of chemicals that play a role in providing part of our defenses. Again, if any of you have taken micro, you probably have spent a lot more time talking about these things in microbiology than we'll talk about them here. And so I know you either got it in micro or are going to get it in micro. So we're not going to go into huge, tremendous depth on these concepts, but we are going to spend a little bit of time talking about them and introducing them. And two of the big examples are two main classes of proteins involved in this second line of defense are interferons and complement proteins. So we'll be talking about both of those in general and how they function.
Dr. Slusky, I have a question.
I have an answer.
You always do. So can we talk about a few examples of first line of defense? Some of the examples they provide us for the skin. So like a paper cut will be a first line of defense, right? And second
so again, great question. If you were to receive a paper cut, then basically what happens is your, excuse me, your first line of defense has been penetrated.
Mm-hmm.
You now have an injury into the inside of your body. And so when that occurs, anything harmful that could have gotten in there will now be attacked by our second line of defense.
Okay, gotcha. And then the other example would be the vaginal canal. So the first line of defense is a secretion, right? And then the second line of defense would be if there's a bacterial infection that changes the pH and the body ends up secreting not so pleasant odor, right? So that would be the second line defense.
Oh, I see what you're saying. No.
I just want to be able to visually walk through. So if there's an infection or injury, this is first line, this is second line, this is third line defense.
Okay, so there's a couple. So again, the vaginal secretions that basically make the vaginal canal acidic are part of the first line of defense, limiting the ability of some harmful bacteria of getting into our body. Okay. If someone were to participate in vigorous intercourse, it is possible that the friction on the vaginal canal could cause an opening to appear in the vaginal canal to pass that non-keratinized stratified squamous epithelial tissue in the same way that a paper cut now has opened the defense that way. So again, that's one of the issues with sexually transmitted diseases. One of the advantages of condoms, they help to limit that bodily fluid to bodily fluid contact that can occur with those types of injuries, I guess, for lack of a better term, that would occur during intercourse. So if some type of sexually transmitted disease was able to enter the female's bloodstream or enter into the areolar connective tissue behind the non-keratinized stratified squamous or something like that, then again, that would be a penetration of the first line of defense that would then require further help in defeating whatever that harmful pathogen would be. And that's when our second or our third line of defense is. My hesitation at the beginning is a change in the aroma of the vaginal secretions doesn't necessarily mean that the first line of defense has been bypassed. Certainly, if there is an injury, that could cause it. But as you mentioned, just a change in the bacterial composition inside the vaginal canal, because those that the aroma of that is caused by the bacterial secretions that are in there, breaking down the glycogen-rich secretions of the female. And so if different bacteria got in there, then potentially as they break down that bacteria, as they break down the glycogen differently, or the other materials that are in there differently, they could produce different byproducts, which could change the aroma that way. That wouldn't necessarily imply that the first line of defense had been penetrated, right? Because there could be something else that could be causing that. Now, again, if there was some type of sexually transmitted disease where there was an injury and it got into the blood, that could also cause it as well. So it just because there's a change in the aroma doesn't mean that there's been some type of penetration of the first line of defense, but it could indicate. So by itself, it's not an indication. It would be an indication that further examination would be required to see what was causing it. But it doesn't in and of itself mean that your first line of defense has been penetrated. Does that make sense? Okay, excellent. All right. Two more important ones. Inflammation is an important part of our second line of defense, and so is fever. So these are the things that we need to talk about now. We need to talk about these characteristics, these processes that help to, again, limit the spread and limit the damage that is done. All right. So the first thing we need to talk about is the mobilization of our phagocytes. Remember, as we talked about in the previous section, in a normal healthy individual, only 2% of the leukocytes are in circulation. So that means 98% of them are housed someplace else. Places like we talked about, like the lymph node, like the liver, like the spleen, like the bone marrow. Areas like that. However, when there's an injury like we have here to the right, then we need to bring those cells out of storage to the site for defense. And of course, as we also talked about, which are the first cells that are going to be involved in this first mobilization?
Neutrophils.
Yeah. So when we talk about this process, wow, my spelling is really bad today. Typically, we are talking first about the neutrophils. However, the same process that we talk about for the neutrophils will also be what would happen with the monocytes with something that was a more chronic injury. So again, if you're just getting, a nail through your finger, the neutrophils are going to be the ones that respond. But if you have some type of chronic affection, the monocytes will be mobilized in the exact same way. And this mobilization is basically a 4-step process. The first thing that has to happen, oops, and I guess I need to, let's do this. Let's make it up here. Obviously, the first thing we have to do is bring those cells out of storage. That process is called leukocytosis. Basically, what happens is our damaged tissue releases chemical signals. It says, hey, I've been damaged. We've been injured. Those chemical signals enter the blood supply. And of course, once they enter the blood supply, where do they go from there?
Site of injury.
Well, they left the site of injury, but once that chemical signal that says, hey, I'm injured, gets into the blood supply, where does it go once it's in the blood supply?
Places where the leukocytes are, like bone marrow.
Yeah, true. Although you guys are overthinking this a little bit. Once something gets into the blood, anything gets into the blood, where does it go?
Everywhere.
Everywhere. It goes everywhere. Absolutely. Right. And you're correct. One of those everywhere are going to be capillaries. And when those capillaries are there, the exchange of materials can take place, and some of those chemicals are going to come out, which means they'll get into the interstitial fluid, which means they'll be picked up by the lymphatic capillaries, which means they could be taken to a lymph node. So notice in this fashion, it can go to the lymph nodes, it can go to the spleen, it can go to the liver, it can go everywhere. And so when it goes to those everywhere places, like the bone marrow, like the lymph nodes, like those, the liver and the spleen, those neutrophils are going to then exit the bone marrow or the spleen or the lymph node, and they enter into the blood supply. However, we have another issue again. Now that our neutrophil has gotten into the blood supply, once it's in the blood supply, where can it go?
Everywhere.
Everywhere. And do we want them to go everywhere?
No.
No, we want them to go to the site of the injury. But luckily, as we talked about, this damaged tissue is still releasing chemical signals. So when a neutrophil gets to a location with a high concentration of that chemical signal, it does something special. And that special thing it does is attaches, let's move this down here. The special thing it does is to attach to the wall of a capillary. In particular, it grabs onto the intercellular cleft. Now, as I mentioned, this damaged tissue is releasing a chemical signal, and here we identified what that chemical signal is. The chemical signal it's releasing is leukocytosis inducing factor, which after you've written it out once, you can abbreviate LIF. So this leukocytosis inducing factor not only tells the neutrophil to come out of the bone marrow, out of the lymph node, out of the liver into the blood vessel, but when it gets to the capillary where the injured tissue is, the leukocytosis inducing factor tells the the white blood cell, the neutrophil, to stay here. This is where you're needed. And so that's what it does. It grabs onto those intercellular clefts, hugging the wall of the capillary. All right, questions on that? All right, excellent. Now, once it's grabbed onto that intercellular cleft, one of the special things, remember, that we talked about with our white blood cells is they have the ability to squeeze their way into and out of blood vessels. And that's exactly what it's going to do. If you've ever seen an octopus, octopi basically able to squeeze themselves through the tiniest holes of things. If they can get a tentacle through it, they can get their whole body through it. And it squeezes its way through. And that's exactly what happens to our neutrophil. Our neutrophil basically squeezes through that intercellular cleft. And when that happens, it then enters the interstitial fluid. And once it enters the interstitial fluid, it is now considered activated. And an active neutrophil is called a phagocyte. So again, while it's in the blood, it is a neutrophil. But while it's in the interstitial, we call it a phagocyte. Think about it, it's kind of like a location name. We've plenty of times in this class given things name based on their location, and this is a perfect example of it. When it is in the blood, it's a neutrophil. When it's in the interstitial fluid, it's a phagocyte. Now, once it's in the interstitial fluid, do we want it to just eat any old thing that it finds? No, we still need to direct it to the, we want it to be selective, and we want it to go to the site of the injury. So not surprisingly, again, we are going to use chemical cues to tell this phagocyte where to go. Do we want it going to healthy tissue?
No.
No.
So healthy tissue actually releases negative chemicals. Repulsive chemicals that discourage the neutrophil, the phagocyte, from going to there. So it's not going to go where those negative signals are. Whereas the damaged tissue is going to release attractive chemicals. So it is a combination of both attractive and repulsive chemical signals that are going to make sure that it doesn't go to where the healthy tissues are and instead comes to the site of the injury. And that process of that combination of positive and negative chemical signals telling the neutrophil where to go, we call chemotexas. And in this fashion, we are able to draw that white blood cell out of the lymph node and bring it directly to the site of the injury, where that neutrophil can become a phagocyte and can start doing its job of gobbling things up. Right, and that's its job. Its job at the site of the injury is going to be to break down dead and damaged cells. That's going to obviously help in the repair process. But what we care about right now is it's also going to break down any harmful pathogens that may have penetrated our first line of defense. Remember, our goal here isn't to keep it out. That's no longer possible. But what we want to do is limit the spread and limit the damage. So if I can get my phagocytes to come to this area and start gobbling up those harmful pathogens, they're not going to be able to spread throughout the body. And if they're not able to spread throughout the body, they're not going to be able to have as big of an effect. So we can limit the damage and limit the effect. Questions on that? All right, excellent. So that is how we get the phagocyte to the site of the injury. What we need to do next is actually talk about what that phagocyte does once it gets there. So again, our first goal was to mobilize. And then once we mobilize that phagocyte and get it there, we then have to do its job. And of course, its job is phagocytosis.
I don't think you're typing.
Oh,
just
you're right, I'm not. Excellent. And so that's what we're going to talk about next. However, before we do that, I think we'll go ahead and take our first break.
I had a quick question.
Yeah.
So in Chemotexas, the attraction and repelling force, it's an electrical force, right?
No, chemical signals.
Okay.
This is, so when great question. When I say positive and negative, I'm not talking about charge. What I'm talking about is attractive and repulsive chemical signals.
How does that work?
The same way that if you had a big, huge buffet in front of you and they blindfolded you, and on one side of the table was spoiled sushi, and on the other side of the table was filet mignon, would you be able to follow the chemical signals and find the right end of the table to go to?
Yeah.
Kind of the exact same way. Some chemical signals discourage the cell from going in that direction, and other chemical signals encourage the cell to go in that direction. And so that's exactly what happens. These chemical signals, some of them basically repulse the cell. It doesn't like them and won't go towards them, whereas others will attract it and lead it towards them. All right.
Got it. Thanks.
Excellent. All right. So let's go ahead and take our first break. It looks like it's 1:20, so we will come back at 1:35. And I will start the recording at that time. All right. Any other questions? All right. See you guys in 15 minutes. So we've gotten our phagocyte to the site of the injury. Now it's going to do its job. Right. As we talked about, these are primarily neutrophils and monocytes, although most of our white blood cells have some phagolytic quality to them. These are the two most common ones. And basically, as you can see, it's a pretty simple, straightforward process. The first thing that is going to happen is endocytosis. Oh, wow, that's massive. Right. This endocytosis is going to occur by the cell is going to attach to the pathogen. Now, how do you think it grabs onto that pathogen?
Recognizes the antigens.
Excellent. Although be careful, remember, this is part of our non-specific defenses. So it isn't that it necessarily recognizes a particular antigen, but you have the right idea. One of the things that can occur is it is able to at least recognize abnormal. Although, didn't we learn about something else that binds to antigens? Antibodies. We haven't talked about the anatomy of antibodies in too much detail yet, but as we know, they're Y-shaped proteins. They have specific binding sites at one end of them, but the tail is a uniform structure. So notice we don't care what it's attached to. These phagocytes, if they see this tail wagging in the air, they're going to grab it. It's just this big flag, generic flag, so it doesn't matter what it's attached to. It can grab onto that antibody and then basically fold its plasma membrane around it and engulf it and bring it inside. And so that is exactly what our phagocyte is going to do. Forms a vesicle around it, and again, as we talked about, we call that process endocytosis. Of course, as we know, anatomists love to name everything. And so when that pathogen is embedded within a vesicle, we call that particular structure a phagosome. So a phagosome is basically, as it says, a phagolytic vesicle. It is a vesicle that contains our harmful pathogen. Now, notice at this point, we have already limited the spread. Right? The first part of our goal has been achieved. By putting it in this vesicle, we can now limit the spread we can stop it from going anywhere else. But we also want to limit the damage that it can do. And so the way we do that is by digesting it, by breaking it down. As we know, one of the organelles that we learned about in 430 was a lysosome. Remember, a lysosome contains a massive number of hydrolytic enzymes. It's a big fancy word. What does it mean?
Breaking down macromolecules by adding water molecule.
Absolutely, absolutely. You absolutely have the right idea. Hydrolytic enzymes use water to break bonds. These are enzymes that break bonds. And so notice what our phagocyte does. Our phagocyte basically takes this lysosome and fuses it with the phagosome. And of course, as we know, anatomists love to name everything. So when our lysosome fuses with the phagosome, it becomes a phagolysosome, which, as I'm sure you can guess, is a word you're almost certainly going to have to spell at one point or another during a lab or lecture exam. All these alphabet soup words are always things that you know you're going to be responsible for. And notice, once these hydrolytic enzymes come in here and are active, then it is able to break down the pathogen, which of course limits the damage. Which is our other big goal of our second line of defense. Now, remember, we talked about these are things like bacteria. These are things like viruses. So they're made up of proteins, they're made up of nucleic acids, they're made up of lipids. Aren't those all things that our body and our cells need?
Yes.
Yeah. So notice, after we break down and neutralize that pathogen, most of that material is just going to be released from the cell. And what do we call that process of releasing materials like that from the cell?
Exocytosis.
Excellent. Into the interstitial fluid so that they can be recycled and reused. However, what was the key word in that statement I said?
Most.
Most, because what does most mean?
Not all.
Yeah. But not all, right? Again, we use that example we were talking about before of the castle in your interactive physiology. If a thief comes to your castle and tries to steal something, what do you do to him? What would they do to that thief?
Try to fight him off.
Right, like Samuel, hammer time, they'd kill him. If you caught that thief, you'd kill him. And then after they killed them, what would they do? Would they just in the middle of the night go out and bury him in some unmarked grave so that nobody was aware of it?
Probably.
Really? Or what did they do? Or if they found a spy in their castle, what would they do to those types of people?
Make an example out of that?
Exactly. They'd put their head on a stake outside of the castle to say, hey, if you come and try to do this to us, do something bad to us, this is what's going to happen to you. And that's exactly what these phagocytes do. Some of the proteins, and not just any proteins, but the antigens of the pathogen, our phagocyte is actually going to take them and express them on the surface. basically what they're saying to all the cells around them are, hey, look what I just killed. One of these was floating around. Because if we're dealing with viruses and bacteria, do they usually just come one at a time? No. So if I found and killed this bacteria, then there's probably others of them around as well. And so by taking this protein and expressing it on its plasma membrane, it is actually going to stimulate our third line of defense, our specific defense. Basically, what this phagocyte's going to say is, I just killed this particular bacteria. So there's going to be more of them around. Go find more of these and kill them. So it sets up the alarm. It sets up that warning to, again, stimulate that immune response to more aggressively destroy whatever that harmful thing is. So not only does it help to limit the spread and limit the damage of this particular virus or bacteria that we've picked up, but any others like it around in the area.
Is that how the mRNA vaccine for COVID works? Like that spike protein is kind of like the head outside of the castle, or is it
Exactly.
different?
You have the absolute right idea. Notice here we are using the antigen from the pathogen to stimulate an immune response. And that is exactly what the COVID vaccine does, the mRNA ones. However, instead of needing to infect you and have your phagocytes break down that pathogen and have it express the proteins, we just give you the messenger RNA, because as you learned in 430, messenger RNA is used for protein synthesis, and we can make the cells make that protein and release that protein. Notice there's no virus involved in it. There's nothing harmful associated. We just are stimulating cells to make proteins, and those proteins, those antigens, are going to stimulate an immune response. It's exactly how it works.
Dr. Slutsky, I have a
Yes.
question. So I just want to go over the semantics. When you talk about the third line of defense, are you referring to the adaptive defenses, or are we still under the category of innate defenses?
Remember, our third line of defenses are going to be the specific. They're going to be the adaptive.
Okay.
Which, again, if you think about it, makes sense. All three of our lines of defenses, do they work completely independently of each other? No, absolutely not. They're going to all work together, right? And again, you may not have thought of it in those terms, but you're aware of it, because when you get sick, what happens to your mucus production in your nose?
It increases.
It increases. So notice there, you've already got a virus inside of you, but now you're increasing the amount of mucus being produced in your nasal cavity to help to, again, eliminate any more from getting in. So all of these work hand in hand. And as we'll see when we get to the adaptive defense, we'll see exactly the vital role that these phagocytes play in starting that process. Yep. So there you go. I've made a mess of the board here, but again, we're going to use those digestive enzymes to break down the pathogen, release most of it as waste that can be recycled. But like I said, some of those antigens are going to be expressed on their surface. And that is what our phagocytes are going to do. In fact, because of that, phagocytes are sometimes referred to as antigen presenting cells. And when we get to our third line of defense, we'll see how important these antigen presenting cells are to stimulate our, as you mentioned, adaptive immunity. All righty. Questions on phagocytes? Excellent. So two processes we've talked about for them. We've talked about how we mobilize them, get them to the site of an injury, and then we've actually talked about the process of phagocytosis, what they do when they get to the site of the injury. But they're not the only cells involved in our second line of defense. Another example of a cell that is involved in our second line of defense is what is known as a natural killer cell. These cells, among with others, but this is the most common type, provide what we call immunological surveillance, basically cell inspection. And I kind of like the analogy that I used before. These natural killer cells are basically the night watchmen at the museum. Typically, they're there by themselves, wandering around in a dark museum with their flashlight, going room by room, looking for things that don't belong. And that's basically what these natural killer cells do. Now, these natural killer cells function in a similar fashion to our lymphocytes. We haven't talked about our lymphocytes, but we'll get there. But in particular, they function similar to T cells. We'll talk about those T cells when we get there. And their job is to basically destroy abnormal or foreign cells. There is a special protein, not an antigen, but a different special protein that is presented on the surface of cells, what is called a major histocompatibility complex. We'll talk about this in much more detail in the next class, but I'll go ahead and introduce them here. These proteins are very important for our immune response. But unlike antigens, these are generic proteins. In your body, you only have two types. So notice we're not looking for specific virus A or virus B or bacteria A or bacteria B. We're just saying, hey, does this cell have one of these two proteins? Check. Does that cell have the two proteins? Check. And so it's just looking for these two really generic proteins. And basically, like our night watchman, wanders through the tissue cell by cell. Are you okay? Are you okay? Are you okay? Are you okay? Looking for something that doesn't belong. And as its name indicates, it is the natural killer cell. If it finds something that doesn't belong, it's going to kill it. Now, by something that doesn't belong, obviously, we mean cells that are infected by harmful pathogens. But as we talked about in 430, cancerous cells typically produce abnormal proteins. So if a cell is presenting one of these abnormal proteins, then it can be recognized as being abnormal, recognized as being cancerous, and can actually destroy that cancerous cell. Of course, there's a flip side from this as well, right? We talked about compatibility issues with blood and how there are two antigens that can cause a severe reaction. And we also talked about how something like the liver has over 200 different antigens and can have as many as a dozen that can cause a severe reaction. So can you take any old liver and put it inside your body if you've been drinking vodka for breakfast, lunch, and dinner for 17 days? No. We have to be very, very careful with our transplants, because if you transplant a organ into our body that a natural killer cell recognizes as being foreign, then it will attack it. It doesn't care the fact that you want it in there, that it's keeping you alive. All that, bumbling, stumbling night watchman is doing is looking to see whether a cell has the right protein or not. And if it doesn't, it can attack it and destroy it. This can lead to transplant rejections, where the body actually fights the organ that you tried to put in there.
I have a question regarding transplant.
Yes.
So what about some of the other non-human tissue, like cadaver tissue or like stents? Like how does the body not attack those foreign objects?
Well, so it really, it all has to do with these proteins that are on their surfaces and things like that, and also where they're located as well. Things like stents or the balloons or things like that that are put in place are non-biological. So they don't have proteins. In fact, for a long time, they were trying to make a artificial blood. Right? Remember how we talked about O is the universal, O negative is the universal blood donor because they don't have those proteins, those antigens on the outer surface. Well, what if they were able to make artificial blood cells out of smooth plastic that didn't have any proteins on the surface or something like that, and be able to use it that way? And so a lot of time and effort went into it. And the problem they found is they couldn't get it to carry and release oxygen well enough. So a lot of it has to do with the presence of these proteins. Your corneas are something that are transplanted quite commonly, and that's because your corneas are avascular. They don't have a blood supply to them. And because they don't have a blood supply, no white blood cells can get there to see that it doesn't belong. So it's much, much easier to get something like that. Same thing with valves. You often hear about people getting pig valves. I know it's ironic because blood's passing through it. But remember when we talked about the heart, the valves are held in place by that fibrous skeleton. And that fibrous skeleton limits the blood supply going to things like those valves. So it doesn't have a big blood supply to it. So again, without blood going to it, we can't recognize that it's foreign, and we won't attack it. So again, it really depends on the blood flow to the structures that's going to affect how and how unique the proteins for a particular organ are that will determine something like that. And I saw that there was another question as well. So you've got the right idea, absolutely. And Amanda did a good job of answering that. Remember, we have that night watchman. When that one night watchman comes across that one cancerous cell, he is able to destroy it. But if you've got a big old huge tumor made up of hundreds and thousands of cells, is that one little Barney Fife going to be able to defeat all of them before they're able to divide and make more?
No.
No. And so, yeah, so unfortunately, they can divide and spread more rapidly than these poor little Barney Fife security guards are able to destroy them. All right. Now, the question then becomes, how do they destroy them? We'll talk about this a little bit more when we get to the T cells, but in general, they're going to do one of two, or maybe both of those two things. And they are either going to trigger apoptosis. Someone remind me again what apoptosis is?
The cell explodes.
It doesn't necessarily have to explode, although that's a good example of it. Absolutely. That's actually going to likely a little more happen with the lysine that's going to happen. What do I actually mean by apoptosis?
It's suicides?
Yeah, basically the cell, it's a programmed cell death. Keratinocytes undergo apoptosis. They fill up with so much keratin that they basically chuck their nucleus, they chuck all their organelles, and they basically die. Notice they don't burst when that happens. They just become dead, flat cells filled with keratin. So apoptosis is a programmed cell death. But you are absolutely correct. The other thing that will do to these cells is they will put special proteins on the surface of the cell that basically pokes a hole in the cell. And if we poke a bunch of holes in the cell, then that plasma membrane is no longer semi-permeable anymore. So what's going to happen? If I poke a bunch of holes in the cell, what's going to happen to that cell?
Water will enter.
Yeah, absolutely. Although technically, if you think about it, if we think back to 430, what's really going to happen, if you remember what really, 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 really wants to get into a cell?
Sodium.
Sodium. So if you poke a bunch of holes in a cell, sodium is going to come rushing in. However, you're dead right. When all that sodium rushes in, what's going to rush in right after it?
Potassium.
Water.
Water, right? Water follows stuff, absolutely. So you're going to have a massive amount of water that is going to come in, filling it up like a water balloon. And when you fill that water balloon up too much, like you mentioned, it's going to burst. So yep, so we can get this cell to fill up with water and burst like a water balloon, or we can get it to basically just chuck its nucleus, chuck its organelles, basically kill itself. Either way, it's not going to be able to cause any damage anymore. All right. Questions on that? All right, so as we see, cells play an important role in this second line of defense, but there are also chemicals that help as well. Let's actually talk first about, let's switch to the whiteboard for this, talk about complement proteins. So again, we're still here talking about our non-specific second line of defense, limit the spread, limit the damage. But now we're going to talk about complement proteins. Complement proteins are a class of plasma proteins, meaning that they're always present, floating around in the plasma in the blood. And there is over 20 of them that have been identified. These complement proteins work in a cascade fashion. What does that mean?
Like dominoes.
Yeah, exactly. I like that, like dominoes. One event leads to the next. And in particular, there are two primary pathways. Your book actually does a great job of describing these in detail. I'm not going to hold you responsible for it in detail. Like I said, I know you'll get this stuff in micro, but we do need to know what the two pathways are and have a general understanding of how they work and how they're different. So let's talk about those two. And we will start first with what is known as the classic pathway. Now, again, remember our goal here is to limit the spread and limit the damage of a pathogen. So I should say in both of these pathways, our goal of our complement proteins are to directly affect the pathogen. So in the classic pathway, for this classic pathway, what makes it the classic pathway is it binds to a pathogen that has an antibody on it. If we think about this, this makes this kind of a directed process. Here we have a pathogen. And as we know, pathogens are going to have abnormal proteins on their surface. Sheet, move this one over here. And in this classic pathway, what has happened is that an antibody has already found it and tagged it. So an antibody is already waving that flag on its surface. And remember, as we talked about, these flags, the ends of the antibodies are always identical. So it doesn't matter what it's attached to. What happens is our complement protein, and in this case, our first complement protein, and guess how they identify our first complement protein?
The tail.
Well, that's how it binds, but what do you think they call the first complement protein?
C1.
Exactly, C1. C1 will come and bind to, oops, the antibody. All right. Now, so again, we have this directed process where C1 binds to the antibody. Once it binds to the active antibody, it becomes active. By that, what I mean, it starts to affect the other complement proteins. When it affects those complement proteins, it, C1, activates C2. And then, as it turns out, C2, with help from actually C4, oops, splits complement protein 3. Again, we don't need to worry about the specifics, but the key to this process is that in this cascade effect, we have this single complement protein that is C3. And with the help of C2 and the help of C4, this C3 is going to be split into 2 pieces. Piece 1 and piece 2. And not surprisingly, they are called C3A and C3B. All right, I'm done with the drawing for now, so let's just talk for a second. Again, this classic pathway is a directed process. It is directed by the antibody. Because an antibody is present, these events occur. First, C1 binds, then all these other things happen. And the key is C3 is split into C3A and C3B. Are we comfortable with that? All right. As long as we're comfortable with that, then let's talk about what these 2 pieces do. C3B is actually going to bind to the plasma membrane of the pathogen. Now, if it's bound to the plasma membrane of this pathogen, can it come back together with C3A?
Nope.
No, if we stick it into the pathogen, then it can't come back together with C3A. And so what that means is C3A is free to float around inside of the plasma of the blood or the interstitial fluid. And C3A basically becomes a strong chemical signal in the fluids of the body. It can attract phagocytes. Remember, we were talking about attractive chemical signals. This can be one of them. It can enhance inflammation, making more inflammation, and have other effects on the cell to basically increase our immune response. So C3 basically becomes, and I'll put a square around it to remind us that this is a protein. Becomes this big chemical signal floating around in the fluids of the body to enhance our immune response, bring phagocytes, increase in inflammation, do things like that. C3b, on the other hand, is attached to the pathogen. So it can be another flag to wave that can be an attachment point for those phagocytes. Notice C3A gives the come hither to the phagocytes to bring them here. And then C3B makes it easier for the phagocyte to attach to the pathogen. But these complement proteins can do some work of their own. It can also be an attachment point for other complement proteins. Again, we're not going to worry about the names of them or anything like that, but let's think about this. A new complement protein comes in and binds next to C3b in the plasma membrane. And then another one binds next to that. And 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 suddenly, what have I done to this pathogen?
Poke a hole.
I poked a hole in it. And if I poke a hole in it, what's going to happen?
Sodium will enter, then water, and then the cell will burst.
Exactly. Sodium enters the cell, water rushes in after that sodium, our pathogen swells and bursts. These special complement proteins that form this hole in our pathogen are what are known as the MAC. The membrane attack complex. So this membrane attack complex forms, and it pokes a hole in the pathogen and can destroy the pathogen that way. So notice both C3A and C3B, once separated from each other, can play a huge role in limiting the spread and limiting the damage that this pathogen can form. Are we comfortable with that?
Professor, when
Yes.
C3b attaches to the plasma membrane,
Yep.
Is there anything it attaches to? How it differentiates the pathogen from normal cell?
The complement proteins help it to do that. So the short answer is it has a special binding site that is able to have attached to the plasma membrane, specifically to the one that it was activated for here.
Okay.
But you do kind of have the right idea, because that's so a question. Any other questions on the classic pathway? All right, if there's no more questions on this, then let's look at our second pathway. Notice this classic pathway is a great pathway because it is a directed pathway. But there is an alternate pathway. Right off the bat, does anybody see a problem or a limiting factor to our classic pathway?
What if there's no antibody?
Exactly. Notice for the classic pathway to work, there has to be that antibody there first. Luckily, our alternate pathway does not require an antibody. However, the downside of that is it is not a directed process. It's actually more of a spontaneous. You could almost call it random. It turns out C3 is not a stable molecule. As such, it constantly breaks apart and comes back together. So if we draw our C3, oops, I don't like that. So C3 will often just spontaneously split into C3A and C3B. But once it separates, like a couple of magnets that I pull apart, they're going to be attracted to each other and they'll go back together. So it'll spontaneously split, and then spontaneously go back together. Come apart and go back together. Come apart and go back together. So just as it's floating around inside of your fluids of your body in the blood, it is just coming apart and going back together, coming apart and going back together. All right, with me so far? However, what if spontaneously, or like I said, arguably just randomly, when that C3 breaks apart into C3A and C3B, what if at that moment of time, there just happens to be a pathogen present? If there just happens to be a pathogen present, then guess what's going to happen? C3B could go back together with A, or exactly, it could attach to the pathogen. Like I said, notice this is not a directed process. This didn't happen on purpose. It was spontaneous. It was lucky. We just happened to be lucky that it came apart at the right time where there happened to be a pathogen present. And now C3B was able to attach to that harmful pathogen. Now, even though this was spontaneous, even though this was arguably lucky, isn't it going to have the exact same effects? Isn't now that C3B is attached to the pathogen, it can be an anchor for that membrane attack complex. It can be a flag that attaches to a phagocyte. Can't our C3A, now that it's free floating inside the plasma of the blood, be a chemical signal attracting phagocytes, enhancing inflammation and everything else? Yeah, once they're separated from each other, they're going to have the exact same effects. It's just like I said, in this case here, it was essentially a spontaneous or random process, right? Which makes it less ideal because it's not directed. But the advantage, as we mentioned before, is that we don't need the antibody for it. Now, I've done a truly amazing job of drawing this. I'll even save it to share with you. But we can also look at the pretty pictures from your textbook as well. So I mentioned those complement proteins, over 20 different plasma proteins. They have that cascade effect and 2 main pathways. Notice here, yes, I will post that picture on the lecture module so that you can have that. So notice here we see the illustration of that classic pathway. Notice again, it starts with antibodies already attaching to the pathogen. Then our C1, first complement protein, binds to it. As I mentioned, it activates C2, then with the help of C3. And again, I don't care that you know the precise mechanism, but notice the effect. The effect is that it is broken C3 into 2 pieces, C3A and C3B. And notice C3B has got that attachment site where it is going to directly attach to the pathogen. Conversely, notice our alternate pathway doesn't require the antibodies, doesn't require C1. Remember, as we talked about, it is that spontaneous process. We just have that random splitting. And if it just happens to split when there's a pathogen nearby, then the special proteins on C3b will allow it to attach to the pathogen. It's not directed, but it also doesn't require the antibodies. So there's some good and there's some bad. But whether it's directed or not, whether it uses antibodies or not, whether, right, it's spontaneous or whether it's directed, the effects are the same. Here's C3b binding, being that anchor point, and notice it causes those other complement proteins to come and form that membrane attack complex that I mentioned, poking the holes and causing it to be lysed. And at the same time, C3A is free floating inside of our humors. As I mentioned, it can enhance the phagocytosis. It can increase inflammation. Now, here we go. This is a good picture that shows it. I like this picture. Let's look at this picture. So again, whether it's classic, whether it's alternative, the events are the same. C3 is split into C3b or C3a. C3b binds, causes the membrane attack complex to form. Again, it makes it easier for the phagocytes to be able to grab onto and hold onto it. Whereas C3a, like I said, enhances inflammation by releasing more histamine, right? It attracts phagocytes. It's that positive chemical signal that can attract the phagocytes to the area. And so these proteins play an important role in helping to, again, limit the spread, limit the damage. Questions on that?
So it all makes sense. One has an antigen, one doesn't the pathogen, but there must be some type of other signaling factor, right? For the pathogen that doesn't have the antigen for this cascade to take place in the alternative pathway.
You're right. The C3b is not just going to attach to some random cell in your body. You're right. It has a special proteins that allow it to bind specifically to the pathogens. Yes. All righty. Now, as I mentioned, complement proteins are not the only chemicals that are involved in our second line of defense. We also have these chemicals called interferons. Let's do this on the whiteboard again, because I like drawing. No, we need a sad cell. Let's make it black. Here we have an infected cell. All right. As we know, in particular, this cell is infected by a virus. And as hopefully you understand the way this process works, a virus red, is going to bind with its protein coat the plasma membrane, right? This is one of the big deals about Delta. The spike proteins that you guys have talked about earlier on Delta make it easier to bind to the plasma membrane of our cells, making it easier to infect it. When it infects the cell, then what it does is it injects a little piece of genetic material, either DNA or RNA. If it's DNA, it's going to go to the nucleus. If it's RNA, it's going to go directly to the ribosome. And basically what they're going to trick the cell into doing is to make new copies of the virus. So rather than making all the proteins the cell normally wants to make, this cell is going to suddenly start making a bunch more new viruses. This cell fills up with viruses until eventually it ruptures and bursts, releasing all those viruses to affect the nearby cells. We comfortable with that process? All right, let's make a happy cell. Oops, no, just a black outline. So that's our infected cell. And that infected cell is sitting next to a nice, happy, healthy cell. So what these interferons do are basically a chemical signal that is released from, where am I going to do this? I didn't leave myself a lot of room. Okay, let's cheat and do this. I'll move that over here a little bit. I can do all my writing over here. A chemical signal that is released from the infected cell to protect its neighbors or warn its neighbors. Again, remember, this is still part of our second line of defense. So our goal is to limit the spread, limit the damage. Notice this first cell, cell A, is infected. There's not much we can do about it. But what it can do is try to protect its neighbors. Basically, it's going to fall on the grenade. I'm a goner. I'm going to die, but I can save you. The way it does this, while it is being tricked into making copies of this virus, it does have some awareness that it is making proteins it doesn't normally make. And so as a result of that, it starts to make some special proteins of its own. And those special proteins are the interferons. It then releases those interferons into the interstitial fluid. So make them little blue specks that are released. And these interferons, well, let's actually cheat and do it this way. Have a special receptor on their surface. So that when our interferon binds to it, it stimulates protein synthesis in the healthy cell. And again, not just random protein synthesis, basically two things. It starts to produce proteins that will either inhibit the binding of viruses and or it will limit replication of the virus proteins. So that when this first cell bursts and releases all these viruses, this virus may try to come and bind to our healthy cell, but our healthy cell has been able to make special proteins that stop the binding of the virus. And if the virus can't bind to the cell, it cannot affect the cell. Or what can happen is that our virus is able to bind. It does release its genetic material into the cell. But again, our healthy cell has been making special proteins, and these special proteins can either stop or limit the production of the viral proteins, which means the cell will either not make new viruses or will make viruses much more slowly. And if we can make it to make them much more slowly, again, we can limit the spread, we can limit the damage. So notice these interferons don't do anything for the first cell. The first cell is a goner, it's already infected. But what we can do is slow the spread of that infection, slow the production of the viruses to limit the spread and limit the damage. And again, we have some pretty pictures from your textbook that show this same thing. Again, interferons are special proteins that are released from our infected cell that bind to the nearby healthy cells, causing those healthy cells to produce antiviral proteins that'll either stop the virus from infecting it or stop the replication of the viral proteins. These chemical signals, again, are released into the interstitial fluid. So again, they can attract the natural killer cells. Right? They can enhance inflammation. Notice a lot of things enhance inflammation. We can activate those macrophages. Tell them, hey, this isn't just a single injury. This is an infection. You guys, we need those macrophages, those big eaters, to get out here and do their job. And again, here's that pretty picture showing the same thing that I showed, right? Our first cell is infected. It's being forced to make viruses, but at the same time, we are able to, forced to make viruses, but at the same time, we are making our own protein signals, those interferons that tell our cell to make special proteins to stop the infection of that virus, or to at least limit production. All right, questions on that? All right, excellent. We are almost done with our second line of defense. We have a couple more things to talk about, and then we can start talking more about immunity and what exactly that means. But I think this is a good point for another break. So let's actually leave it on the whiteboard this time. We will take another 15-minute break. It looks like it's 2:35 now, so that means coming back at 2:50. And we will start the recording at that time. All right, any questions before we take our next break? All right, I'll see you guys in 15 minutes. All righty. Next big major part of our second line of defense. Again, our goal here is to limit the spread and limit the damage. And how does inflammation do this? Well, what are the cardinal symptoms associated with inflammation?
Redness.
Swelling.
Pain.
One more.
Heat.
There we go. Excellent. Why? Why does this all occur?
Histamine.
Excellent. So there you go. This process is driven by chemical signals. Again, more chemical is involved in this process. And one of the main chemical signals that is responsible for this process is absolutely your friend and mine, histamine. What cells again have and release histamine that can drive this response?
Basophils.
Excellent, basophils. And of course, where are those located?
In the blood.
Yeah, that's not trick. Wasn't a trick question. In the blood, excellent. What else? What other cells have we talked about in this class or all the way back in 430? Why might we have talked about that also contain a large amount of histamine?
Dendritic cells.
There you go. Oops. Where were those dendritic cells, the cells of Langerhans, located again?
In the skin.
Yep, and more specifically in the epidermis. Excellent. And there was one more, and I'll give you a hint. It's found in areolar connective tissue. Anyone remember the cell in the, say again?
Mast cells.
Mast cells, there we go, excellent. Those mast cells in the areolar. So again, these and other cells are capable of releasing chemical signals. Damaged tissue is able to release chemical signals, but histamine is one of the major ones that lead to these four cardinal symptoms. Excellent. But what actually causes the redness? And actually, maybe we should reorder this a little bit. What actually is responsible for causing the redness and the heat?
Rush of blood.
Yeah.
Exactly. One of the things that histamines and these other chemicals do is they dilate the blood vessels. And by dilating the blood vessels, they bring more blood to the area. As we know, blood has hemoglobin in it, so that causes the reddish coloration, and it brings heat. Because remember, our blood is 2 to 3 degrees warmer than our core body temperature. What about the swelling and pain?
Leaky capillaries.
Exactly. Histamine doesn't just dilate the blood vessels, but it makes the capillaries more leaky. As a result of that, our capillaries are more permeable. As a result of that, we get more filtration. And as a result of that, we get a swelling, we get edema. Oh, shoot. Can you guys give me one quick second? Pause this so we don't have to stare at me doing this. Do that. So we get that edema, and that edema pushes on the nerves. And that causes the pain. So basically, these two main characteristics of our histamine are what cause these 4 cardinal symptoms. And ultimately, though, it comes back to that original question. Why is all of this important? Why have the area become red and heated?
Well, if you get more blood to the area, doesn't that allow for more defense cells, whatever they may be?
Yeah, more white blood cells to the area. And also, if it's all those red blood cells causing the redness, why might that be important?
So that we know something's wrong.
True, although one of the other things on the list does that. Redness, why is the blood red?
Iron,
Yes, iron
And
material.
what's binding to that iron?
oxygen.
Yeah. If we are injured, aren't we going to probably want to heal that tissue, and won't oxygen and other nutrients play an important role in that process? Yeah, so not only are we bringing more white blood cells to the area, but we're bringing more red blood cells to the area. We're bringing platelets to the area. Right, all these things that can help in that process of stimulating defense. Let's talk about the other thing you mentioned, right? What's the advantage of getting all that swelling? What's the advantage of the edema?
Can't it help like protect, like give extra, I don't know, cushion around an injury or something?
Okay, excellent. I like that. That's good. Absolutely. It does. That swelling around the injury can provide protection. But remember, also think in terms of lymph nodes. If we have a bunch of fluid rushing and staying in that area, that edema causes congestion. And as we know, congestion slows things down. So if we can congest that area, is it going to be easy for any pathogens that got in there to spread to other parts of the body?
No, it'll be harder for them to spread.
Yeah, it's going to be harder. And as you guys talked about, that pain makes us know that it is injured. So we're going to protect it. If you injure your ankle, you're not going to go running 5 miles on it, right? So the pain lets us know so that we protect it. We don't use it. And again, if I'm constantly moving my arm, I'm increasing the circulation of that. And that can cause more spread. If it hurts and I'm protecting it and I'm holding it still, then again, that's going to limit the blood flow. We've got that congestion in the area, and it's going to limit the spread. The good news, I think most of this stuff is pretty intuitive. You know, we're all kind of familiar with inflammation and its influences and its effects. So there's only one minor issue. Are we going to get away with terms like redness, swelling, pain, and heat in a class like this?
No.
No, of course, we're going to need to use the appropriate anatomical terms. And as usual, the appropriate anatomical terms are in Latin. So, of course, the terms we will use on the exam are rubor, calor, tumor, and dolor. And of course, what is what? What does rubor refer to?
Redness?
Redness. Calor?
Heat,
There you go. Tumor.
swelling,
And so then that makes dolor.
Pain.
Pain. There you go. Excellent. So again, histamine and other chemical signals. We talked about that. I love this picture. I've had this picture forever. I don't even remember where it came from anymore. But if you think about it, it kind of really illustrates what's happening with inflammation. We have an injury, right? We have a splinter that has gotten in, causing our mast cells and other cells to release histamine. Chemicals from the damaged tissues are released, and their effect is to cause the blood vessel to dilate. Notice our blood vessel gets bigger. Notice the intracellular clefts become larger. So our blood vessel is getting bigger. Our blood vessel is getting leakier. Notice, and we didn't even talk about it, but when you make those capillaries more leaky, when you increase the size of the intracellular clefts, it makes it easier for complement proteins to come out. It makes it easier for phagocytes to come out. Those complement proteins can tag our pathogens. Our phagocytes can gobble them up. So as we talked about, these different systems work together. So this is a great illustration of showing this process of inflammation and how it helps with other things to, again, limit our spread, limit the damage that is caused in this second line of defense. Now, like I said, I don't have any idea. I don't remember. I've had this picture for so long where it came from, but your book has a pretty picture that shows this process as well, where again, shows that redness, that heat, that swelling, that pain, that is part of that, that is part of that response to the injured tissue. And again, when we're talking about inflammation, we're talking about this side over here, driven by histamine and other chemical signals. All right, that leaves us with one more important non-specific second line of defense, and that is fever. Fever is basically a way that we're able to reset that thermostat. Like very first physiological process we talked about in 430 was the importance of maintaining a stable or relatively stable body temperature. But with a fever, we want to increase that. This is primarily done by the releasing, again, of more chemical signals. In this case, certain leukocytes release a special chemical signal called pyrogens. When they're exposed to a pathogen, they release this chemical, and this chemical basically does a couple of things. One of the things is it resets our thermostat, making our temperature go up. This helps to limit, to not only increase our body temperature above 99 degrees, but also helps to retard sweating process. Because as we know, sweat and that water is very effective at bringing the temperature down, right? One of the things that they talk about is when the fever is broken is when you get all sweaty as a result of that. And that moderate fever is incredibly beneficial. Iron and zinc are very important for certain viruses to be able to replicate. So when we increase our body temperature, those excess minerals can be stored in places like the liver and spleen, limiting the availability, again, making it harder for the pathogens to spread. But just the temperature itself has a huge effect. Just raising your body temperature 1 degree Celsius increases the metabolism of your cells by 10%. And so the harder the cells are working, right? The stronger the cells are working, the faster the cells are working, the better we're going to be at fighting off whatever that harmful pathogen is. Of course, that means if a little fever is good, then a big fever is even better, right?
Of course.
No.
Of course, exactly, absolutely. No, of course not. And of course, the reason for that, what makes high fever so dangerous?
Because you,
I'm sorry, go ahead.
Do you need your proteins?
Yeah, exactly, right? As we know, proteins are highly sensitive to pH changes and temperature changes, right? And as we've talked about, every single chemical reaction that has ever been found in the body. Again, in A&P, we rarely use ever, we rarely use never. But as far as I know, there is not a chemical reaction that takes place in the body that they have not found an enzyme that can make it faster. And so if you increase the temperature too high, you can start to denature proteins, and that can dramatically affect the function of the enzymes, causing major, major problems. Which is, of course, why when you have that high fever, the best thing to do is to go jump into that ice cold bath, right?
No.
No. Don't we need to bring the temperature down?
Yes, but if it's too fast, we go into shock.
Absolutely. And not just shock, but there are other issues as well. One of the important things to remember is that not only is it the high fever that's dangerous, but as you guys have absolutely correctly pointed out, rapid changes in temperature can be dangerous as well. And not just coming down, but also up. Absolutely. Shock can be one of the things that can occur. That's a little bit more common in adults. But in kids, especially in infants that are, there's the magic 10-10, around 10 months of age or 10 kilograms of weight. If they have a rapid spike in temperature or a rapid drop in temperature, at that stage of development, their neural system is not fully developed. Their neurons, their axons are not fully myelinated, and it can actually cause a neural storm, right? Causing a febrile seizure. Febrile seizures are actually quite common, much more common than we expect, and they are terrifying when they occur. Or at least terrifying for the parents who haven't experienced before, right? When the EMTs show up, they're very casual about it because, again, they deal with probably, my guess would, several a week of these that are occurring. But a febrile seizure is different from a seizure in an adult. Typically, it is not indicative of any long-term neurological issues or anything that long. It's like I said, it really has to do with these rapid changes of temperature and their effect on the nervous system. Where the baby will basically go into this shaking fit. It may seem a little bit of extreme, but the baby will contract its muscles, will shake a little bit. Typically, they're very exhausted, very upset by it after it occurs, not nearly as upset as the mom is after they've occurred. But like I said, there are no associations with them and long-term problems, neurological problems. But like I said, they can be very scary when they occur. So it's not just the danger of having that high temperature, but rapid changes up and down. So you don't take the baby and you put it in ice water, right? You put them in a nice warm bath. The febrile seizures only happen in infants, that is correct. But as someone mentioned, the body can go into shock, the heart can stop. There can be other issues that can happen from rapid changes in temperature. So you don't get into that ice cold bath, you get into a lukewarm shower, a lukewarm bath to be able to slowly bring the temperature down, right? In ancient times, one of the things that your great-grandparents may have done to your grandparents is they would take alcohol and, like rubbing alcohol and basically put it on a washcloth and rub it over the surface of the baby. It evaporates very, very rapidly and it causes a rapid change in temperature. Of course, the problem in doing that when great grandma did it is that alcohol can be absorbed in through the skin. And so you could actually get the baby drunk. It's even more severe if you do it now, because the difference between rubbing alcohol now and rubbing alcohol in your great grandma's day is now rubbing alcohol is denatured, right? The problem was, especially during Prohibition, people would drink rubbing alcohol. It doesn't have a good taste, but it would get you drunk. Right. So what did they do? They denatured it so that your body is no longer capable of taking advantage of the alcohol that way. So now when you put rubbing alcohol on their body, you actually are poisoning them and they can die. So don't do those things. Sure, absolutely. So something like that can occur. You also see that in some of those cryo centers, where people will get the ice treatments and then, you know, go from the hot tub to the ice treatment or go from working out to the ice treatment and back again. Yeah, those dramatic change in temperatures can affect blood pressure, can affect heart rate and things like that, and can cause someone to pass out. Absolutely. That one's not neurological. That one more has to do with the physics of the function of the heart and cardiac output and those things that we talked about. But yes, so the take home message is A, don't give your kids alcohol, and B, again, gently bring down those temperatures, not rapidly. All right, questions on any of that? All right, your book's got some great examples of some of the things that we talked about. Oh, urine, there you go. That was one we didn't talk about for our first line of defense. All right, the acidity of the urethral tract is important for protecting that that pathway to the inside of the body as well. Our second line of defenses that we've talked about. What we are going to talk about in the next class is going to be our specific defense, our third line of defense. Again, what is important about this one is that this one is precise. This is that scalpel defense. This one, not only does it care whether it's a virus or a bacteria, but it can tell the difference between virus A and virus B. And the way it's able to do that is the antigens. The precision of this system comes from the antigens. This allows this system to be very, very versatile, so that we're able to use it for all sorts of different harmful pathogens. It allows it to be very precise and very effective. And it is systemic, right? When I cut my hand and my hand swells as a result of that, does my foot swell as a result of that as well? No, those natural killer cells, right? They can only roam so much because they're just one cell. Those interferons help the local cells surrounding them, but they don't help cells on the other side of the body. But the key to this third line of defense is it's systemic. It can go everywhere. So to find those viruses in all your nooks and crannies, and probably most powerful of all, not only does it help you for this exposure to the pathogen, but it has a memory, allowing you to respond to that pathogen if you're ever exposed to it again. And that memory not only allows you to respond a second time, but you are going to respond faster and stronger. This sounds amazing. Why do we need the other two if we have all of this?
It's slower acting.
Exactly. The one downside to this process is that it is a slower process. Right? When you have that hammer, you see something move, you hit it with a hammer. If you have to wait to see precisely what it is, to know precisely how to take care of it, that's going to take a little bit of time. And so often, the downside of being slower is that often we will have symptoms. That pathogen will have the ability to have some type of effect on us before we're able to efficiently get rid of it. Now, like I said, we're going to talk about our third line of defense in the next class. But before we do that, and of course, like I've already said, they work hand in hand. Before we do that, I want to talk a little bit about immunity. I've used this term when I got on my soapbox that we don't have an immune system, we have an immune response. But we need to talk a little bit more about what we mean by immunity. Because immunity actually means a couple of different things, right? We have, like I said, this third line of defense, our specific resistance, our specific immunity. And actually, let's switch to the whiteboard for this. when we have our specific immunity, and actually let's do it this way. Specific immunity comes in two flavors. The first is what we call our innate immunity. Innate immunity is basically your genetics. This is what you were born with. And actually, let's say it that way. Innate immunity is what you're born with. And obviously, this is determined by genetics. What you got from mom and what you got from dad. It's a very incredibly interesting area of study. They actually did a study many years ago where they put a bunch of men and women in a room together. And what you would do is you would have a conversation. A man would have a conversation with a woman. They would talk for 5 minutes. And at the end of the 5 minutes, you would smell the person's neck. And then you'd move on to the next person and talk to them for 5 minutes and then smell their neck. And at the end of the experience, it's kind of like a speed dating, exactly like a speed dating. And at the end of this speed dating experience, they then rated the people on how attractive they found them. And then they ran their genes. And what they found is that people were more attracted to people whose genetic makeup was more different from their own. Why would that be the case?
To make better babies.
Yeah, more diversity in your defenses. Again, we haven't really talked about antibodies yet in great detail, but we know there are these Y-shaped proteins that have these fancy shaped binding sites, squares and circles and triangles. And what determines whether you have all three of these, or maybe you just have squares, or maybe you just have circles, or maybe you have star shape or rectangles or octagons? What determines the shapes of these binding sites?
Genes.
Yeah, your genes. Your genes determine your starting point, right? Maybe you have relatives that came from South America. So you happen to have a gene that produces a special antibody that is capable of binding to malaria. But if you never get any further south than, Merced, are you going to have to worry about coming in contact with malaria? No, in fact, you may go your entire life living to 140 years old and never come in contact with malaria. And this antibody will never be used. But you still have it because you got it from your parents and your grandparents and your great-grandparents and so on and so forth. So this innate immunity is what you are born with, and it is determined by your genes. Does that mean if you don't have that malaria antibody, you're just screwed?
No.
No, because we also have an acquired immunity. Does your acquired immunity have anything to do with what you were born with?
No.
No, this is due to what you were exposed to. Right? This is determined by your environment. You remember back to like the 50s and 60s, one of the big questions that science was all worried about was, is it nature or is it nurture, right? What determines who you are as a person? And of course, we now realize that is an incredibly stupid and simplistic argument because clearly it's both. And we see this here in immunity as well. Obviously, your genetic starting point gives some people more of an advantage over others by having more diversity, more different types of protections. But we have that ability to obtain more, to acquire that immunity. All right, so when we talk about our specific immunities, we have that innate, which is an important starting point, and then we have acquired. However, is there just one type of acquired immunity? Well, I'm asking the question, so what should the obvious answer be?
No.
No. There is active acquired immunity. With active acquired immunity, you are exposed to the antigen. As a result of this, your body does the work. Now, like many things, there is a positive and there is a negative. What is the positive to your body doing the work? Your body makes the antigens, the antibodies.
You have an unlimited supply?
No, well, true. You can produce a tremendous amount of a massive amount, enough of them. But remember, the other thing we talked about with this adaptive immunity is you get the memory cells. And remember, those memory cells allow you to produce a more aggressive, a faster, a stronger response. So much so that the next time you're exposed to it, it's so fast and so strong, you don't even have any symptoms. But what about the first time you were exposed to it?
You'll get the symptoms.
Yeah. Downside of this one is that you do get symptoms as a result of it. So when we're talking about our different types of acquired immunity, we have this active acquired immunity. This is when your body does the work. You make the antibodies, you get the memory cells, but it takes time, right? This is a slower process. And because this is a slower process, you also run the risk of getting the symptoms associated with it as well. And not surprisingly, because I've left a lot of room on this screen, there are two types of active acquired immunity. There is a naturally occurring active acquired immunity. What's happening here? Oops. Well, remember, you're exposed to the pathogen. You're exposed to the antigen, I should say. With naturally occurring active immunity, how do you think you're exposed to the antigen?
Just naturally through your environment?
Yeah, you are exposed to the pathogen. You come in contact with the pathogen, right? In your environment. All right. As we just finished talking about, there's a big negative to this, right? When we're actually exposed to the pathogen, we get sick. All right, we can have symptoms. But like we said, the positive part about this is we do the work. Because we're doing the work, we get a strong memory cell. And that strong memory can protect us if we're ever exposed to that ever again. This right here, when we use that term immune response, oops, oh, I don't know what I just did. I don't like it. There we go. When we use that term immune response, this is really what we're talking about. When we talk about our body's immune response, we are talking about that natural occurring active acquired immunity. However, as we know very, very well, we can also have an induced active acquired immunity. What's going on with induced active acquired immunity?
Like a vaccine or something like that?
Exactly. Notice this is what happens with vaccines. With a vaccine, you're not actually exposed to the pathogen. Oh, I guess technically you can be. You are either exposed to one of 3 or now 4 things. You're exposed to either dead pathogens. In rare instances, they will give you very weak pathogens. But as we also know, we can just expose them to the antigens. And now we can actually expose you to the messenger RNA, which causes you to make the antigens. So I don't even have to inject you with the antigen, the protein. I can have you make the protein for me. Huge positive benefit to this. What is the huge positive benefit to this?
You don't get symptoms?
Exactly. No, there is no, and again, I will say no here. Again, there are a few, they're not rarely, they're not commonly used anymore, but obviously if someone uses a weak pathogen and you have a compromised immune system, there is a small chance of you getting infected, but almost no, let me rephrase that. None of the common vaccines use weakened pathogens, with the exception of some of the flu vaccines can sometimes use weakened, but it's not nearly as common. But you've got the right idea. I'll answer your question, Maximilian, in just a second. There is no or almost no chance of actually being affected. Right? If there's no pathogen, you're not going to get sick. It's impossible. And so you're right. The COVID vaccine doesn't have the virus. It is impossible to get sick from the virus because the vaccine doesn't have the virus in it. It has the proteins that are going to stimulate the response. Right? We used to use dead pathogens or weak pathogens before we understood how it was that we recognized them. But now that we know it's just the proteins that we recognize, we don't have to run the risk of getting infected. We can just give you the proteins or now have you make the proteins yourself so that there is no chance of being infected. Does that mean that everybody feels completely rosy after getting a vaccination? No, obviously it will still stimulate an immune response, which is really bad, right?
Not necessarily.
No, that's exactly
Might
what
feel
you
bad, but it
want.
doesn't do anything.
Yeah, that's exactly what you want. You want to stimulate the immune response. So you may get some achiness, you may get some soreness, you may get some swelling, you may get a fever, because what you're doing is tricking the body into thinking that harmful pathogen is there. So your body does the work, right? There's no actual risk of infection, but your body still does the work. And what's the advantage of doing the work?
Then you have the antibodies when you really need them.
Well, not just the antibodies, but you have those memory cells that we talked about. You get the memory, which can protect you the next time. All right. It's one of the ridiculous things about the anti-vaxxers saying how, you know, the vaccine, the COVID vaccine doesn't stop you from getting infected. No, of course it doesn't stop you from getting infected. No vaccine stops you from getting infected. What it stops is the severe reaction, because your body is able to respond to it more rapidly and more strongly. And the data shows that. Those people who were vaccinated and get infected typically have very minor symptoms. And the number of people who die who have not been vaccinated versus those who die that have been vaccinated are, you know, astronomically different. Now, doesn't mean it's all rosy. What's the negative? I mean, aside from the microchips that, you know, Bill Gates is putting into our bodies, what's the other negative besides the microchips?
That's a positive.
It will make it easier to talk to your iPhones. What's the other, is there a negative?
Some people can have like allergic reactions, but that's more situational.
It is a little bit more situational. And also a lot of that has to do with either eggs or mercury or things like that, which are almost never used in vaccines anymore. So the
Kind of
chances
like
of that,
byproduct type.
yeah. So yeah, that could be a potential, but that wasn't actually what I was looking for. Although I'll put that down. However, I will remind you that the potential reactions to Advil, right, acetaminophen, ibuprofen, those types of things are just as, if not more severe than for vaccinations. But what's the other downside of vaccinations? What's the big debate going on with vaccinations right now?
Sometimes it will affect the body that cause death.
So no, the vaccine itself doesn't cause death, but the big debate that's going on right now has to do
Temporary.
Like the reaction of the body.
True. So again, and I think that's where some of the confusion comes in. You want the body to react. That shows the body is doing the work. Yes, some of the potential is that it could wear off, but guess what? It can wear off also from the naturally occurring. So that isn't part of so much the difference. The difference I was thinking of is the fact that they're looking
Mandating
at
it.
boosters,
Oh,
right? Because while it is stimulating a response, typically the response to just the antigens is let's say it this way, causes a weaker response, right? You're not getting as sick, you're not doing quite as work, so it may require multiple shots, or it may require boosters to get the same full effect that you would get from actually the vaccine itself. And again, this isn't something that is new for COVID, right? Back in ancient times, and by ancient times, I mean when I was in elementary school. When I was in elementary school, Timmy down the street got chicken pox. So what did all of our parents do?
Kept you away from Timmy.
Did they?
They got you
they took you
infected.
to Timmy's house so that you
No,
would
they
get it.
actually took all of our parents, packed us all up, took us to Timmy's house, rubbed us on Timmy, had us hang out there at Timmy's house for a couple hours. We had a chicken pox party. Everybody on the block all had chicken pox at the same time. All right. So why did parents do that?
Because you would get shingles as an adult, which is way worse.
True. And also for males who have never had chickenpox and never been immunized for it, if they get it as an adult, it can actually affect their reproductive system and they can become sterile as a result of chickenpox. So not only is it bad for both women and men for shingles and things along those lines, but also it has a much more severe effect on adults, especially males, than it does on kids, right? So we got the chickenpox party, everybody got chickenpox, and everybody lived happily ever after. right? My kids have never had chickenpox, but what have they had?
The vaccine.
The vaccine. And how many of those shots did they get during their academic careers?
I think it's just one or two. One in a
I think
booster.
it's at least 2, and it might be 3. Isn't it 2 for chickenpox? And then maybe a third. I don't remember exactly, but often not mumps, measles, mumps, and rubella. Many of these vaccines, you have to get multiple shots at certain times apart, again, to increase the number of memory cells to have that same effect. All right. Questions on that? It doesn't make sense now. It's just going to get worse because we're not done here. All right. No questions. So we have active acquired immunity. So let's take a step back. Guess what else we have?
Passive.
There you go. What's passive acquired immunity?
No ATP?
So normally you're right. When we think of passive, we think of active and passive, we think in terms of ATP. That's excellent. And you absolutely have the right idea, right? Notice in active, we may not have talked about ATP, but we did say that the cells were doing the work. Well, in passive, the cells aren't doing the work. Instead, with passive, what actually happens is that you are given the antibodies directly. Notice in this case, your body does not do any work. Is there a positive to that? Well, I know we haven't talked about the process yet, but do you think the body is able to instantly make antibodies?
No.
No. Remember, as we talked about, it's a slow process. It takes time. So if you're super, super sick, do I just hope that you're going to get better by making your own antibodies, or should I inject you with some? Yeah, the advantage of this is it's way faster. You get that effect of the antibodies immediately. However, is there a downside if you're not doing the work?
Yes.
Yeah, no memory cells. So the advantage of faster is typically you're going to have fewer symptoms or less severe symptoms because it's going to be that much faster, but no memory cells. So that means that if you're exposed to it again, you're going to have to deal with it again. And since there's naturally acquired and induced active acquired immunity, guess what? There's the same thing for passive. What do you think an example of naturally occurring active acquired immunity would be? Oh, sorry, passive acquired immunity would be.
A fetus.
Say again?
A fetus
True. How is the fetus getting those antibodies?
via umbilical cord.
Yep. Via the placenta to the fetus. Anything else?
Breast milk.
Yeah. Or breast milk to the baby. Absolutely. All right. So again, like we talked about, the advantage of this is that it is going to be fast. Right. This is especially important for that fetus and the baby because their own immune response is not developed yet, or at least not fully developed yet. But like we talked about, the negative, I guess A for advantage, P for positive, N for negative, is again no memory. So mom's protecting it, but that's a good thing. Where mom is, baby is usually. So what mom's exposed to, baby's exposed to. So mom's body can do the work and provide that extra protection for the baby. And of course, if there's naturally occurring the passive acquired immunity, there must of course be induced passive acquired immunity. What would that entail?
Injection of antibodies for an infection.
True, and it doesn't even have to be an infection. Probably one of the best examples of this is snake venom. Antibodies can bind to the snake venom, stopping the spread of the snake venom and limiting the damage that that snake venom can do. However, snake venom is very, very dangerous. So do you want to wait till your body can build up its own defenses for something like that? No. So you get bit by a snake and they give you an injection of an anti-snake venom. Of course, if you're a snake wrangler and you get bit again the next week, what happens?
The response is less severe.
Is it really? If I'm giving them the antibodies, are they doing any work? Are they building up any defenses?
No, it's just the same.
Yeah, so it's just the same. So every time they get bit, they're going to need another shot. Now, you are partially right. Maybe the second time they get bit, or the third, or the fourth, or the fifth, or the 10th, but maybe by the 12th or 15th time they've been bit, then maybe they might start to be able to have built up their own antibodies and their own memory cells slowly. But if you're a snake wrangler that have been bitten 15 times, you might want to find another job. So for the most part, again, really, the only difference between these two is how you're getting the antibodies. The effect is pretty much the same. It is fast. So as a result of that, you have fewer symptoms, less damage. But again, the negative is the same. If you're not doing the work, then no memory cells. So you don't get no extra protection. Right. So really, for these two, the passive immunity, the induced and naturally acquiring, it's really the main difference is how you're getting the antibodies. I guess the only other difference is with an injection like that anti-snake venom, right? I'm not just drinking mom's breast milk where I'm getting a broad spectrum of antibodies from mom. I'm injecting the very specific antibody for that specific, that for specific problem. So I guess the one advantage to the induced, the additional advantage to this induced is more specificity. Because you can just give them the antibodies for whatever it is that they're dealing with. Notice I've made a very pretty flow chart here, but your book has this exact same thing. Again, we talked about the innate and the acquired immunity. For that acquired, we talked about how there's active and passive acquired immunity. For active, we have induced and naturally acquired. And for passive, we have naturally acquired and induced. So again, we have that same flow chart that I made here. And the nice thing about my flow chart is we emphasize the positives and the negatives, the advantages and the disadvantages to these. All righty, questions on that?
Professor, I have a question about the active acquired immunity. So if you got exposed to the pathogen and you get those strong memory cells, so what's the point of the vaccine at this case? So what I'm trying to say, if you, for example, got a COVID, what's the point of getting vaccine if you
So
already have those?
the one answer that I would give to that, and again, that is something that is being debated. The argument for the additional vaccine is twofold. The first, as we've mentioned, the more you're exposed to something, the more memory cells you're going to produce, the more defense you're going to build up. So the stronger the defense you're going to have. Right. It's one of the reasons why, again, they're even looking at a third booster now for people who are immune compromised. If you're immune compromised, as you're being exposed to these things, you might not be building as many memory cells because you may not have as many cells to produce them to start with. So the more exposure you have, the stronger the defense you're going to be. The other advantage comes from the specificity that we were talking about. This is a scalpel defense. It is very, very precise. It's one of the reasons, for instance, we need to, go ahead and put this off the screen. You need to get a flu shot every year, right? It's not like you get one flu shot when you're 10 and you never have to get a flu shot again. The problem with the flu vaccination is the flu pathogen, that virus, mutates. And if the proteins change shapes, then our memory cells can no longer recognize them. And so we lose that advantage of the memory cell. And that's why we have to basically get flu shots every year, because they're constantly changing. Chickenpox is much more stable. Its proteins don't change. So that's one of the reasons why its vaccine is so effective. You get it as a 9-year-old and you basically never have to worry about it again. The advantage of the COVID vaccine is more variety in the shapes of the proteins that are being made. So it can give you a broader defense for those mutations that appear to be occurring. I mean, the fact that in a year and a half, we have gone from the primary to now a Delta, and there's been other ones that have already been discovered. I know Mu is shown up. Where was Mu coming from? Colombia? I don't remember. There are a couple of other forms that may or may not be as contagious as the Delta is, but so depending on when you got it, if you got it from the original, if you were originally exposed to it, that original strain is almost non-existent anymore. So the variations and the differences in the Delta may be something that could be beneficial because especially Moderna, it's shown is incredibly effective against the Delta variant. So the flexibility of the vaccine gives us a more wider range for potential variants that may be more effective that way.
So the memory cells you get from that first COVID is not going to be helpful for the Delta?
It depends. Well, it may, but it may, again, it depends on how much it's changed, right? So you may not get as vigorous a response. And so that's why getting a boost with something that is more similar to the Delta may then be able to provide a more wider defense against a second exposure to the COVID.
Okay,
Yep.
Thank you.
Any other questions? All right, awesome. This went a little longer than I wanted, but we still have 45 minutes left. So again, I'm going to break us up into our groups now. I'll put you guys in your breakout rooms. So I need to bring up my, where's the list? Here it is. All right, I will break you guys up into your groups. And then again, you're welcome to use this time to continue to work and discuss. If you have any questions from me, you can either come back to the main room or there's a way to flag me down and be able to do that. Let me hold on while we're here. Let me go ahead and stop the recording because we don't need that anymore.